For the structural comparison, we will look at the structure of B-cell receptors and T-cell receptors side by side. Here on the left side, I have shown BCRs. And on the right side, there are TCRs. Let's start with B-cell receptors, or BCRs. B-cell receptors are the membrane-bound immunoglobulins, consisting of 4 polypeptide chains. Among them, 2 are called heavy chains, and the other 2 are called light chains. They are named based on their molecular weight. And the chains in the BCRs have both variable domains, with diversity in amino acid sequences, and constant domains, with similar or constant amino acid sequence. In each light chain, there is one variable domain, presenting as VL, and one constant domain, named as CL, whereas, in each heavy chain, there is one variable domain, VH, and 3 constant domains. We named them as CH1, CH2, and CH3. Both these light and heavy chains are connected with each other and also within themselves by a special linkage called disulfide bond. And these BCRs are similar in structure to the secreted antibodies, but there is one difference: that the membrane-bound immunoglobulin, or BCR, contains 2 extra domains. One is the transmembrane domain, which anchors inside the cell membrane, and the second one is cytoplasmic domain, which transmit the signals to the cell cytoplasm. Thus these 2 domains are additional in structure of BCR than the secreted antibodies. Now, let's look at the other side. On the other side, the T-cell receptors, or TCRs, are heterodimeric proteins that are composed of 2 proteins or polypeptide chains. And in most of the TCRs, these protein chains are alpha chain and beta chain, while few are made of gamma chain and delta chain as well. Each chain consists of a variable, V region, and a constant, C region, where a variable region is a portion with a variation in amino acid sequences, enabling them to bind with plenty of microbes. And this region is named V-alpha in alpha chain, and V-beta in beta chain. And the constant region is the one with similar amino acid sequences. And this region is presented as C-alpha in alpha chain, and C-beta in beta chain. A portion of these constant domains are anchored inside the cell membrane, making its transmembrane region. And just like BCR, both these polypeptide chains are connected by disulfide bond. Now here, again in BCRs, the variable regions of both the heavy and light chains form a special site called antigen binding site, which exhibits tremendous diversity in its amino acid sequence, and thus allows BCRs to recognize and bind a wide range of antigens. Whereas, in TCRs, the variable regions of both alpha-beta or gamma-delta chains form this antigen binding site for the recognition and binding of antigens. But in TCRs, there is only one binding site, whereas in BCRs, there are 2 binding sites. There is another interesting fact related to this binding site. Well, in TCRs, this antigen binding site recognizes and binds to the antigens that are presented by the special molecules called major histocompatibility complex, or simple MHC, found on antigen-presenting cells, or APCs. But in BCRs, there is no such requirement, as the antigen binding sites in BCRs are able to bind the compatible peptide antigens directly, without the need of any antigen MHC complex. Opposite to this binding site, there is the site from where these receptors anchors over the surface of their respective cells. In BCRs, this site is named base or FC portion. That is made of the constant domains of both heavy chains. There is no involvement of light chain here. This site helps them anchor over the surface of B cells and are involved in signaling. So, these BCRs are expressed on the surface of B lymphocytes, which are responsible for the production of antibodies and humoral immune responses. Whereas in TCRs, this site opposite to antigen binding site is also named as base, but unlike BCRs, it is made of the constant domains of both alpha and beta chains. This base helps them anchor over the surface of T cells and is involved in intracellular signaling. Then, like BCRs, these TCRs are expressed on the surface of T cells, which are a diverse group of immune cells with various functions. These T cells are then involved in cell-mediated immune responses, including the activation of other immune cells and direct killing of infected or cancerous cells.
Discover a wealth of accurate, up-to-date medical videos for in-depth learning, anytime, anywhere. Elevate your medical expertise with sqadia.com.